The overall goal of this method is high-throughput parallel analysis of multi-member microbial communities in confined environments using silicon microwell arrays.
The main advantage of this technique is that it allows for a high-throughput, highly parallel screening of finely localized microbial interactions that are significant to industry, medicine, and the environment. This method can help answer key questions in the biomedical and microbial ecology fields, such as how do spatial constraints inherent at fine scales drive deterministic and stochastic parameters of community development, and what are the key effects on microbial member abundance and organization.
Begin with preparing the microwell arrays. First, cut individual microwell array chips from silicon wafers printed with multiple arrays using a diamond scribe. Each individual chip contains subarrays of wells with diameters ranging from 5 to 100 microns at 3 different spacing densities. On each chip, the complete motif of wells is also repeated 4 times. Next, make a humidified chamber using a pipette tip box and a PBS-soaked wipe. Then, apply a 150 microliter droplet of BSA solution to each chip and incubate the chips in the box for an hour at room temperature. Meanwhile, prepare the bacteria. Spin down a culture and resuspend it in 500 microliters of fresh R2A medium with 2% glycerol. Then measure the culture density at 600 nanometers and adjust the concentration to an optical density of 0.02. After the chip incubation,